So yeah, good afternoon and thank you so much for joining. And this is the Environmental Biotechnology Network webinar series. So just to straight up acknowledge the sponsors, that's the Biotechnology and Biological Sciences Research Council and Engineering Physical Sciences Research Council of UKRI. And today we're actually co-badging this seminar series with the National Biofilms Innovation Centre. So yeah, we're very grateful for the support that's been given so far by Will Green and colleagues within the NBIC to help launch this new series. So the aim of this seminar series is quite straightforward. It's to disseminate innovations and research into water biofilms, and with the prospect of setting up a EBNet, Environmental Biotechnology Network working group, which the vision is to establish a group of multidisciplinary researchers with interests in managing and engineering water biofilms. So if you are interested in joining that working group, please do pop me an email. And just to link it back to the work that the NBIC are doing in this space, so they've got these 4 strategic pillars, if you like, of managing, preventing, engineering and detecting biofilms. And this isn't just in water, we should acknowledge this is in lots of different matrices that might be relevant, so clinical settings, the environmental sector, ship hulls, etc. So lots and lots of applications of how to prevent, engineer, detect and manage biofilms. Now, within this particular working group and seminar series, we're focusing on perhaps 2 key topics within that. So that's how to engineer biofilms for useful purposes. So that could be for treatment, it could be energy recovery, and also managing biofilms within drinking water distribution and storage systems and wastewater conveyance points. So this is a sewer network. So why might we want to do this? Well, you know, we've got two images here. We've got a corroding sewer, so we've got an aging asset base. We get excessive biofilm growth, which might lead to things like microbial induced corrosion, poor microbial water quality through leakage. But there's also opportunity to not just manage this, but also engineer this through things like in-pipe treatment. And certainly that's an emerging area within drinking water distribution, but also sewage conveyance systems. So we've got opportunities here to help manage and engineer these systems. But this leads into water and wastewater treatment as well. So we need to achieve the same levels of treatment without increasing carbon, energy or the entropy of these treatments. We've got to better manage all harmful emissions. This includes things like nitrous oxide, which is a big concern in particularly biological forms of wastewater treatment. And then we've got to manage things like emerging contaminants, viruses, plastics, forever chemicals, etc. One of the topics for today, which I'm really pleased Henry's going to talk about, is energy recovery, but also things like nutrient removal as well. So all of these processes use biofilms. We need to get to ultra low nutrient consents, but we need to do this in a way where we're not significantly altering the water industry's ambition of getting towards net zero by 2035. We need to achieve energy security, and I think this is a topic highly relevant at the moment. And then, as I said earlier, we need to better manage our waste. So the 2 confirmed speakers we have at the moment, Dr. Henry Lee, who's going to be talking today, and Professor Joey Boxall from the University of Sheffield, who's going to be talking in our September version of this seminar. And I should just say that if you are interested in speaking at the Water Biofilm Seminar Series, please do get in touch with our amazing admin team, Louise and Ange at EBNet at ebnet.ac.uk, or myself, and we are available on Twitter and LinkedIn should you need to get in touch in other ways. So we are looking forward to a packed schedule over the next 12 months or so. So without further ado, I'd like to hand over to Dr. Lee.
Okay, thank you. Thank you for Francis and Cranfield College for the invitations. I didn't know this is the first one, so I really feel honored for the first kickoff talk. So today my topic is granular activated carbon biofilms for methane productions from wastewater, and I want to address a little bit about this, how we can do non-classical, energetics. So this project actually has been doing more decades. So this is working with Stanford at the United States. So we have some slide from them, and we share some sort of samples and analysis together. So we'll have some scientific understanding, but also we're going to have a pilot scale study kind of result to share with you. Okay, so I'm going to that. You know that in 21st century, we have a lot of problem, energy security, resource shortage, and climate change, and so on.
Sorry, Henry, you're not sharing screen yet. I would, yeah.
Oh, okay. Let me. It's okay now. Is it okay now? Can you see the screen?
Yeah, that's great. Thank you.
Okay, great. So yeah, I'm just talking about this topic about granular active carbon biofilms, and this project with Stanford together, Chuanqi. And because of first one's century, we have a lot of issues, especially climate change, and especially in UK, we are talking about net zero greenhouse gas emission by 2050. So how we can convert the waste into energy is also one of a big issue. So this is not a trivial test. So I usually talk with my students, with my US cooperators, say we're looking for special microbes, special biofilm microbes, like adventures, so they can do some super power, bacteria power to do wastewater recovery as energy. So we are how we can seek Goldilocks condition for overcome the classical bioprocess limitation, special energetics. So I'm going to talk about this. So today I want to get an analog about, there's one called interspecies direct electron transfer for energy manipulation. So it's really similar like Dr. Marvel's, Captain Marvel's, they can manipulate energy through her superpower. So today I'm going to talk about how we found this and how we can implement in full scale, even pilot scale application. So you might know that energy, electrons, can be act as particles and wave. So the classical one, we usually talk about wave functions, motion. But however, if you want to understand, can we get a better result for energy recovery? There's a way called wave functions that is basically the quantum mechanics, that if we can manipulate the wave function of electrons, can we capture more energy? Can we get a faster through the bacteria and recover as a methane, as an end product? So that's the idea. So there's one quantum mechanics called quantum tunneling. It sounds pretty bizarre, but in reality, it's in showing our world, like in your mitochondria, you do quantum tunneling to capture energy through your life being. And also leaves, they do quantum tunneling to capture photons for harvest energy. For this is how we life turns out. So we can see that quantum tunneling means that there's a wave function of energy, and the heat up green is kind of an energy barrier. But you can see if the wave function is quite good frequency with this energy barrier, they turn through. And when they turn through, the speed is faster than the original one. So this is really bizarre. And why is that? Nobody knows. We just observe that, but nobody knows how to do it. So we are engineering. Maybe we don't need to dig out into the reality or theory, but we can think how we can apply this in this quantum tunneling for energy capture. So the thing I want to talk about, the concept is all kind of reaction has a barrier. That's called activation energy. So this line is so-called catalyst, but still you have a kind of small active energy. So if you have a organic, you want to produce methane, you need to kind of like this here, okay? However, if we go through tunneling, it can be a no barrier or less barrier, so you can get a better kinetics limitation. The other thing about thermodynamics is that if your barrier cannot make it through, if you can turn it through, you can have better reaction. So you reduce the energy level for product, so you capture more energy. So today I'm going to talk about those two, kinetics and thermodynamics, and this example. Okay, so what I usually do is that for understand that, so I do thermodynamic calculations, stoichiometry, and then we use genomic to understand the protein, DNA, RNA, and so on, what kind of pathway inside. And then we look at how the electron can turn through, look at the protein level. Then we apply into the full scale application. So this is the whole stuff. Well, we're gonna cover it for today for those methodology. And the other thing is because it's a wave function, is quantum mechanics. Sometimes it's difficult to calculate using our classical computer in that. So in Imperial, we have a Center for Quantum Engineering Science and Technology. So I'm a board member for that. So we use a quantum computer simulates some sort of wave function in terms of electrons. Okay, so thank you for this center. Now I'm going to introduce one technology, so-called granulation active copper with a membrane. So we are talking about sewage wastewater. So we use this carbon GAC as a carrier to create biofilm, and later on we have a membrane to kind of do separations. So the idea is how we can produce methane through it. So you can see the this is the Stanford, my colleague, we're working together. So we have a process that first stage, we have a active carbon. So this fluidized membrane bioreactors, and with a membrane in the second side stream. So then we use biogas scalding effect for fouling control. But the thing is, we have a really create a good biofilm for a bacteria to survive here. So you get a good permeates, and you can have a GAC biocarrier inside. Then the good thing about the GAC, they can retain the long SRT, okay, for the biofilm, and the membrane can get a really good effluent quality, okay, and also they can retain the solids, so have a good hydrolysis removal, and then you can decouple HR SRT in different way. Of course, you have HRT differently, SRT for MLSS, that's one you do hydrolysis. And then for micros, you can do a little bit methanogenesis part, so that you can decouple SRT in this way. Right, so you can see the batch continuous lab scale result. So we have GAC, so non-conductive material is a control, and conductive material is a GAC, and also we have a carrier coating with conductive material together. You can see the COD remove in the kinetics increase quite quickly. You can pick up very quickly, and methane increase production rate so quickly, and also for that you can see the methane yield increase. Why is that, and how it turned out this way? So this lab scale, and so you can see the reactor that. So we do a little bit understanding about what is a electron with so-called biological quantum energy. So this is fluidized reactors. You can see that. So we can see that one ATP formation, it taking about 20 kilojoules per mole substrates. However, in this reactor we calculate, it can lower as 12. Okay, so by some simulations, and we are not going into detail, but I'll have more later on. And then we found out the acetates and propionate can lower down 0.5 milligram per liter of propionate and acetates, which is not very common in such a low strength wastewater. Okay, so that's what we observed through the study, and we run it for lab scale and also in the pilot scale. We also find this result. It's quite bizarre, too good to be true. So then, yeah, the question is that the most of the biofilms, because we control MLSS, so most of the mixed liquor, or say bacteria, actually is in the GAC. Okay, about 90% of biomass. We do some sort of a take sample and understand how much is biomass located in. So 90% to 80% in the GAC. So we get the extracted DNA and RNA. And then we found out, you can see that. So this is a little busy diagram, but I want to just point out this is a pilot scale system in the summer winter in Korea. and this was in a lab scale. And at that time, we studied in Korea because it's very cold weathers. We also have Stanford one, also Hong Kong one as well. Then you can see that we look we just forgot about hydrolysis for the moment. We look at the Central, the propionate to acetate. You can see this guy, Semaphila, has a lot. Okay, so we found out the kinetics in low strength of this wastewater switch, and I just want to point out this is raw switch and this is lab scale. Okay, we just give a propionate and acetate. You can see that this guy, Semaphila, has a good abundance. And then we have a good methanotrix, both in pilot scale and also lab scale, as in high abundance. Yeah, you can see that the only one stand out, methanotrix. And the interesting is that we found out geobacters also in high abundance. The result here, actually, it was 7-8 years ago when we found out it's quite bizarre. Why geobacter in inside? Because it was just microbial cell, usually dominant system. But usually those two guys are competition in microbial cell in that sense. But then we have come up with a hypothesis that would those two guys cooperate together and how? So then we did an even in-depth genome building sequencing. You can see that we got a really good quality. We got a read, short read and long read peptide together. We got a good methanotric consia. This is the guy who has high abundance, and we got a really good high quality of methanogens being, and of course others as well. So we are quite interested in methanogens, how they take up a cooperation with the geobacter. That's our hypothesis. Geobacter lovely. Okay, so in high abundance in both lab scale and pilot scale. So we recover and look at pathway in geobacter lovely. Yes, you get all of the acetate to CO2 and also electron out. So this guy, geobacter, consume acetates and then shoot out electrons. And then we just find out that the active carbon actually is the conduct materials. So they give a lot of excitements and also uncertainty that why they can capture electron like this way. And you can see this metatranscriptomics result. So all the gene expression are really, really good, regulated with the electron shooting out. And then the methanogen, yes, we're also quite interested about methanogen, this species. And they do uptake acetate. So from our previous understanding, acetates is they compete with the geobacters on this substrates. Of course, they have a pretty good gene expression, but at the same time, we look at another one. If the electron shooting up with the methanogens can take over the electron and get CO2, yes, it also highly expressed. And this methanotrix doesn't have the pathway for taking up hydrogens. So the only way they can take up is acetate. The other one, it will be possibly in DIET diets, get the electron out from geobacter through GAC and take over to methane. The other interesting, also this RNA metatranscriptome result also show really good gene expressions, but in compared to previous one, yeah, so it's pretty high expressed. So it means that some methanotrix using acetates, some using direct electrons from geobacter through GAC. So they have some sort of ratio percentage, but this upregulated result cannot quantify how much methanogen use acetate, how much go with the electrons. Okay, but you can see it's already highly regulated. The other thing we also find very interesting is that, oops, sorry about this slide. I blocked too. Sorry about that. There's another one called Ribisco pathway that they can use CO2 as electron donor, and then it's like leaves. So I should remove this out. Sorry about this. So this pathway is new. So previously we talked about acetate and CO2 reduction through electron. The other also possible for direct electron transfer is go through reversal pathway. So this is like photosynthesis, a little bit differently, but they could go through this pathway and also produce their self-synthesis and also methane. So this is really, really interesting. And we found out they can use up NADH. Okay, so this can be a possibility that NADH is produced by this cell envelope through the GAC tunneling. So this gives us some hypothesis how the electron can capture. Okay, then we kind of come up conclusions that a lot of people also report the DIT that the acetate can be used by DIT diets and then produce methanes. Okay. And then I want to also share with you about my colleague and Stanford. They also do some calculation on the acetates of VFA into methane. We found out this, of course, VFA is very important for methane productions. And the potential minimum effluent for VFA concentration we call S minimum. So it means how low of the substrates can be utilized. So they did some sort of minimum substrate concentration calculations. So this uses as a minimum. So then you can get K value and decay. Decay will be fixed, but the specific maximum growth rates. I know that there's a lot of people do AD1 modeling, so you probably know that. So this two value give you kinetics, and this one also give you another way called thermodynamics. How much the energy is low enough you can capture. So this is Chu-Huang just mentioned. So he did simulations. There's Yeaton's papers. They did this methanometric. This is the methanometric calculations. So they find out the K value is this, and then mean value is that. So it's quite high. So it means that they require longer time for kinetic reaction. So that is the one. And then they require a very high of S minimum, means that this is the effluent quality for our sewage. So it's really difficult for this system, this bacteria actually, can meet the minimum requirement for sewage effluent quality in terms of COD or BOD5. Okay, so it's really hard to meet a secondary effluent quality for BOD standard because the S minimum is so high, 21. So that's why we reported in our textbook, like anaerobic sewage treatment would not work for sewage. But then we got a data, and then we found out with a low concentration of COD, it can be possible. The difference is the methane and COD ratio is quite low. Okay, we'll talk about this, why, and we have the data to share with you later on. So this is a comparison of those same methanometry. We just find out you can see the kinetics rate increase dramatically. The utilized ratio is really high compared to the AD process, and this is with the GAC membrane. And you can see the S minimum, maybe thermodynamically, is much lower, okay, compared to this one. So they give us a little bit concern why this biofilms with the GAC work quite well. So this is the idea we are looking at, especially in methanometrics. So the ideas will go back to the tunneling part. So we have this GAC as a conductor materials, and the electron assume it can tunnel through to methanometrics. And how this possible? Would that be feasible? Then we look at the GAC. We look at their the pore size and tunneling. We found out the size is really small. It can be possible to create a really good electrical density to look at inside. So we did a few of our students we have from Oxford, and we called together. They are physics students, and they are quite familiar with the quantum tunneling. So idea is that we have a methanogens like here. So this is GAC, and we found out it's possible that they might have some sort of conduct material such as quantum sensing and so on, but we don't want to talk about that because we didn't measure the quantum sensing and so on, but we just use water as a media, would that be possible? Because water is the one of the feasible way to do tunneling. And then we want to hypothesize, how about water? If the water worked, then other quantum sensing, other conductor material biofilm might work mostly. So that we will come with a worst case. So we use the water layers as a wave functions, okay? And the water, if we use, it's a polarization of molecules. If you have a really good density of the power, like power density or electrical density, they can be polarization become stick, okay? Of course, it's smallly linked up together. And then they can do tunneling, okay? So that is the idea. So we look at the methanogens S layer, okay, and their compound and so on. We did theoretical calculation. Is that possible? And I want to point out is that there's one references from United States, University of Nebraska. They did pure methanogens. They have a methanol methanosacina, and they want to find out the tunneling effect on the methanogens without any material, just pure methanogen. So there's one material called methanophytosine. So methanophytosine is actually here. It's intermembrane substance. It's also conductor material. So they calculate how quick of this electron transfer through methane productions. Is that quantum tunneling? So they figured out, yes, it's in a range of 1 to 1.5 nanometers. They do tunneling. Okay, so actually most of the methanogen do the tunneling. It's just we don't really look into that, okay, in detail. Okay, but our understanding is that in our system we use a similar ideas because we have a GAC and we have how much of the geobacter abundance, and we fit in COD about 100-ish, 150 row switch into the system, and we calculate the GAC, the power density, and how the water can be a media for tunneling, and what is the minimum range of that they can reach. So we found out the methanogens consilia, this one, and Barberry is about 30 nanometers with the sewage. It can tunnel through. Okay, so it gives a little bit theoretical understanding is possible. Okay, so this is my quite encouraging why we see such a good kinetics rate and thermodynamic rate. Okay, so that is the theoretical understanding. So now I'm going back to the pilot scale study. So the previous data, the Chu-Huan calculated those kinetics result as minimum and K and mu value as from the Korea. So that is a pilot scale they are working on. And now they have one more in the Stanford, been operating 5 years, 20 cubic meters as well. And there's another one new one called SMBR 2.0. So this is the result I'm going to share with you, because this process also compare with NBR systems. So I'm going, this is Chu-Huan again. So what we have to do is that mostly Chu-Huan did was that he want to compare the CSTR and row membrane, and then the one with the GAC. So this is a stop system. So I want to take you the picture. So they have a pilot scale study in parallel fitting a sand sewage from Stanford. And then you can see the first reactors, they have a GAC right here, and they have a membrane. And then they use a biogas to do the gas scalding effect to control the fouling. And the other one without a GAC, okay, just normal anaerobic membrane bioreactors. Okay, so we want to understand the startup periods, footprints, and energy efficiency and CO2 removal. So you can see that this is a micro screen. So they have a row switch, and they don't have this kind of primary segmentation. So they use a micro screen. So this micro screen is a 2 millimeters screen. So actually most of solids that will be retained, and then maybe think about putting into anaerobic digestion. So you capture more solids for anaerobic digestions, and then you have a more soluble or fine fine particle into this one is a safe MBR, which I mentioned now with the GAC one and the membrane. And this is 20 cubic meters. And this one, the conventional one, a little bit smaller, 5.5 cubic meters. And then we want to do head-to-head comparison. So we can see that the HRT for this GAC type of reactors is actually is right here, blue colors. Okay, starting up, and then the influence all the same, all in this because it's a kind of parallel. But I want to let you see that the result for effluent quality. Okay, so the GAC one is really good. They can up to about 30 milligram per liter, lower than that. And then the CSTR is not as good as the GAC one. So we can see that the head-to-head comparison with the GAC additions really play a very important role on the effluent quality. Okay, so I also want to share with you about the energy balance. So Chu Huan also did quite a lot of energy balance in the systems. So he found out it's based on the COD mass balance, and then also 98 some assumption 98% of dissolved methane recovery, and then about 40% of electrical energy conversions from CHP. So he found out this net energy positive is possible through this safe MBR with the GAC in there. So this is quite encouraging in the sense that we are looking at. Okay. And I also want to share with you a really interesting results we found. It was also in Korea sample that we found out we also have head-to-head comparison with activated solid system. So we are talking about this pilot scale. So the yellow one is a pilot scale system, and we look at different disinfection byproduct and pharmaceuticals. So the blue one is inference. So okay, and then we have different pharmaceutical tests, and then we have the full scale activated sludge systems in compare with the pilot scale of safe MBR. You can see that most of the super pharmaceuticals been removed in the systems. Okay. And we are actually doing some sort of in-depth analysis to understand what's going on, because some people also say that it can be absorption by the GAC, and then that probably by the absorption. But I want to point out is this system, the result we found is measured by also Stanford and Berkeley. It was running by about 2 years, and then we take a sample, grass sample, and then we found out really good performance. And we have the whole genome now doing in lab analysis with my student at Imperial, and we do find quite a good pathway capture from genomic analysis, but the result haven't really published yet. We're still working on manuscripts, so it's quite encouraging. So the hypothesis would be it can be absorbed in the first, but because we have biofilms, then their bacteria slowly kind of eat up those pharmaceuticals as well. And quite interesting thing is that we just report the geobacter here with acetate, right? And methanotrix, they do DIET. But we also have sort of a hydrolysis bacteria and other fermented bacteria. We also find a genome bank has this PELI, electron transfer mechanism. So we still analyze it, and we still try to map in those pathways concisely before we can make a really confirmed conclusion. But I can say that 90%-ish, we really think the hydrolysis also happen in this DIT with the GAC part. Okay, so it's give a little bit new insight for information that this GAC, very simple thing, you add in the systems, and it create DIT, and then possibility that the electron can be exchanged by turning through rather than by this conventional process with particle process. Oh, and also one thing I also want to share with you is that because some of the uncultured species, they also have a good metabolism for remove pharmaceuticals. So we think about maybe this directly electron transfer can enhance those hydrolysis because it creates a more energetic possibility for this hydrolysis microbes to do the job. And you can see that those acetate hydrogen and formates, those intermediates, they diffuse those intermediate by diffusions. But if we do while this kind of electron transfer through GAC, it's kind of speed of light, and really quickly you can get a really good efficiency. And also some possibility of tunnel through make it impossible or degradation pathway, make it right way to do happen because you surpass energy barrier. So that is something we're actually doing together with Stanford and some colleagues in the United States and Germany group as well. Okay, so this idea. So I guess that is pretty much I have for today. I make a very short work, so I would like to have your questions and discussion with you. So my summary is that it's possible that if we have a good kind of understanding material, what if it's conduct material into our reactors, where the bacteria can kind of enhance tunneling, and then we can get a better result. Okay, so that is pretty much I have for a short talk, but I really would like to engage with your Q&A. Okay, so this is the project we're working on. So we started with the INHA and Stanford's, and then other group from different world like Hong Kong, Oxford as well. That's pretty much I have so far. Okay, thank you.
Great. Thanks so much. Just if you could mute your microphone. Yeah, fantastic. Fabulous. So a very interesting talk about approaches to potentially boost the productivity of our biological processes. So with that, I'd just like to open questions to the floor. So if you could introduce yourself, turn your camera on, mic on, that would be great, so we can start the discussion. Any questions? Okay, well, I'll start. We've got one on the chat. Fantastic. So David Inman says, do you need to replace the GAC in your system, and if so, how often do you envisage you might have to do that?
We in the Korea one, they operate 5 years. We didn't replace anything, but we sometimes saw that's GAC losing by when you look at the mixed liquor. There's some blackish stuff, but in 5 years you don't replace anything. That's the how long we run in the Korea one. For the Stanford one, it's been just running 2 years. Both of them are in 2 years, so I haven't replaced any GAC yet. Okay.
And presumably the membrane might help retain the GAC in the reactor.
Yes.
Great. And in your system, just a quick one from me. You mentioned that the decay rate in your kinetic modelling was fixed. Do we think that assumption is so it was on your kinetic slide, Henry? Yep.
I'm going to here. This one.
Yeah. So just talk us through that. So do you think that is representative of what we might expect in reality, or is it just one of the assumptions that you have to make to?
Okay, so this one is calculated by Yeatons, like 1992. So this work is we get the full scale GAC biofilms, and we put in bottles, and we make this BMP tests and do those calculations. So this is quite real number, and that. But we also did another study for this kind of column type of lab scale. We also find a really good kinetics. So I'm going back to that reactors. Sorry about this. So we also did quite a lot of kinetic study this systems, and we measure each columns of VFA, and it's really interesting that the VFA into level is up to 0.2 milligram per liter of propionate and acetates. Usually you don't get this low VFA, and we was quite surprised on that mechanism. So we did those candidate first and do genomic later on to figure out what's going on. So we got a lot of challenge in the beginning, but because of genomic workout, okay, that's a diet. And also recently people also kind of report as a diets. And then, yeah, this one a bit more kind of, people were kind of, oh, that they might be possible in this sense.
Fabulous, thank you. I'm going to ask my colleague, Dr. Suniti Singh, to unmute her mic and turn her camera on so we can facilitate a more active discussion. Otherwise, it would just be me and Henry having a chat. She could always say no. Yay! Hi, Suniti.
Hi.
You're on mute.
Automatically mutes. Can you hear me?
Yes.
Hi, I'm Suniti. I'm a postdoc at Cranfield University. That was a great presentation and on a very, very interesting topic. So I was curious about, you use GAC, but there are so many different kinds of GAC, even from like say commercial manufacturers like Norit, they have different kinds. So was it a certain kind of GAC that you used, like with respect to pore size, surface area?
Yeah, this one, I forgot the brand name. Yeah, I used to have it. It's from States. It's not like special GAC, we just use it. And it's usually they use for drinking water filtrations. That's it. And the size we use about 2 millimeters to 4 millimeters. And the fluidization actually we use because we don't want to take a lot of energy. So we use a more like gently mixing. You can see that they have very good columns. They're very clear liquids. It's like I also create a biofilm. The key one is I think is how you operate in gentle movements and create a biofilms. That is difficult part. But so GAC, I don't see that is quite challenging. I also read some paper they use graphene and different kind of material. It also work for the DIT diets. But most challenging part is how you fit into your pilot scale, right? Fluidizations and fluid mechanics, it's a key.
Do you think if there was another kind of media used, would it affect the performance?
Yeah, we also try plastic one, lab scale, with the coating with organic conduct material. It also works. You also have a high abundance of methanogens, that species. However, the geobacter species will change. I think it's related to the conduct material difference with that kind of P-like shape, P-like structure, sort of thing. But methanogens really seem very, very kind of strange that it always there, stick without GAC or with a different kind of conduct material. That's what we found. Yeah, very interesting results.
Thank you. I would just keep on asking some questions, so I will let other people ask other questions. It's a great presentation. Thank you.
Thanks, Suniti. Great. We've got one on the chat from Geraldine Shortland, who's asked, did the lab or pilot scale have fixed or variable flow? As you know, is there any anticipated effects of variable residence time on the performance of these conductive material systems?
No, no. For example, the pilot scale, we use 5-hour HRT, so it's fixed, and you can see the inference is variable all the time. I'm going to get to the slide very quickly. Sorry about this. So we have this 3 kind of different period time of pilot scale. We always fix the HRT. Okay, so you see the in the beginning it's a bit long HRT because we want to grow the biofilms, but then probably 3-4 months we can hit it about 5 hours. So it's quite promising. Actually, we also did in the Korea the temperature really low. We are talking about -10 in the atmosphere temperature, probably lowest one about water temperature about 10 Celsius. It still performed quite well in a sense.
Right, so even at low temperatures, it was working well.
Yes.
Did you have to seed the reactor with anything, you know, to get it started, or
No, which you use the AD sludge and sit it directly.
Okay. So you did need a sort of an inoculum from a pre-existing reactor.
Not from the activated sludge system. I mean, just from the AD sludge.
Yeah, from an AD sludge system. Yeah.
Not from the. You need to kind of specialize those biofilms. The key is how to operate, how you maintain the fluid mechanics, and how you do gentle mixing.
Great. So we've got a couple more questions.
Thank you.
Fabulous. Sorry, did Jay have a question there in the chat? We've got a hand up.
Got Sarah?
Sarah?
Yeah. Hi. Hello. It was a nice talk. I think most of your research work is more aligned to our company research at Waste Bristol. It was really interesting to get more insight analysis, and it's pretty good, like how GAC is more influencing the methane production. It was really a fantastic, really fabulous results. We're just curious to ask questions like, do you have any of the control experiments without GAC to compare the methane production with and without GAC stuff?
Yeah, so this is the one I just mentioned. So we have this systems pilot scale. It's also a membrane without GAC, but smaller scale, and we can do head-to-head comparison. But you can see this one, the cross orange color is the Evan quality. So the GAC one has better one. So we also have the lab scale as well. So we also find that with the GAC had the best performance. And in the beginning, it's difficult to let people believe. But because we'll do scientific understanding, so it kind of match up quite well. So I assume that maybe GAC might be not the best non-conductive material. So we also think about maybe some researchers also can find a plastic one with a coating conductive material. It will also work. We also have a few publications that it works as well.
Oh, thank you.
Fabulous. Thank you, Sarah. Appreciate the question. One from Professor Sonia Heaven at Southampton University, who said, fabulous presentation. We've tried running similar GAC anaerobic MBR systems with dairy wastewater, but had trouble establishing good performance. Any insights as to why our process might not have worked quite as well as yours?
Okay, I guess the most challenging part is to create a biofilms. How you can gentle mix things and don't get clogged. If you have a high concentration of that, I don't know if there's a lot of solids inside. So the one we get is we get a micro stream. So in the pilot scale, we get a 2 millimeters of quite small particles. Of those particles can get into the switch, as a safe MBR system. So I guess is that if you can control most of the soluble one and also get a really good kind of recirculations, and that might work better in our understanding.
Great, thanks, Henry. I mean, do we think that the substrates themselves in the sort of dairy wastewater, long chain fatty acids, etc., could be contributing to that? Yeah, compared to municipal wastewater as well.
I think so. If you have more soluble substrates, I think it worked quite well. Yeah, but you probably have to be a little bit beginning with a little bit slow down, like longer SRT, build up the biofilms, and slowly move to your target HRT. So just build up that kind of biofilms is really key informations that you can reach your goal.
Fantastic, thank you. Was there a legacy hand there from Sarah, or is that you got another question?
Oh, sorry, that's the old hand. Sorry, I've taken it.
No problem. Fabulous. Sonia says thank you. Yeah, I was just a follow-up question really. So if from Suniti's question, so if we were trying to sort of take this work further, we could either try and optimize the pore size distribution of the GAC or the particle size itself to give us more effective surface area. Did you see any sort of clogging of the media, of the GAC? Because traditionally we'd use GAC in a drinking water works, and usually pretty far to the end of a drinking water treatment works. So it's very interesting that you've applied it for municipal wastewater here.
Yeah, like the first reactor in pilot scale in Korea, it did clog. So that's why we use the mesh, like micro mesh, to create the problem. So the second generation, third generation quite well. If you can remove most of particulates, and it will be fantastic. So mostly we want to have a more soluble, substrate getting the systems. So that might be get a better one, because hydrolysis is so difficult, most likely in the sense that how we can repeat. But it also makes sense. You can separate solid and liquid more, and you also get a more AD digester sludge, just do more methane on that side. And then you get a separate in the more liquid part, use short HRT. So you can have a shorten the footprint. So you can have a pretty balance. The other thing we also just want to let you know that our nutrient is also one of the things, because this doesn't remove nutrient at all. So I think the best way is just recovery. We might use a chemical process to recover ammonia and phosphorus. And we have some idea, but still maybe some people in the industry or academia, maybe that's also one of the things that you may be interested in that. The other drawback is a lot of dissolved methanes. It's oversaturates because we get a really short HRT, and somehow the methane dissolve into these liquids. So that might be another burden. So we try different way to recover methanes, especially in Stanford team. They do like cyclone, just create different gravity, or they can vacuum out, can get some sort of method. But easy way, they even try using bubbling, put the air, and then you can flush out the methane. But how you can recover those methane gas form, and then send to CHP is also another kind of problem. So, and hydrogen sulfide is also one of the things. But we found that if we use some chemical reaction to recover phosphorus, some hydrogen sulfide also can be pulled down together with phosphorus. So that might be also new challenges ahead of us.
And any sort of data on not just the the amount of methane, but the quality? What's the sort of balance with the methane versus the CO2 in the gas?
Oh, the gas, pretty good methane concentration, because usually CO2 is dissolved into the liquid as well. So you get a pretty good high concentration of methane. And actually in the Stanford one, I guess they have a 500. You can see that 500, 600 kind of COD. They got a really good methane composition. I remember it says 70 or 80. That's really high. Yeah,
it's pretty good.
Yeah, that's high concentration of COD feed.
Thanks, Henry. We've just had a sort of follow up from Suniti on her experience with dairy wastewater. So startup time took a very, very long time with these wastewaters, and it's linked to excessive washout and low growth rates on the biofilm. Angela Bywater's just mentioned that you ran the system, Henry, at lower temperatures. What was the range of temperatures that you operated the system at?
I just mentioned in Korea it can up to 10 Celsius water temperature, and it worked. And Stanford one probably lowest one 15 or 18. California, the weather is really nice, so it will be no problems with that temperature. For the dairy waste, I also remember that the Stanford also cooperate with the Swiss. They also have 2 pilot plants on running dairy waste. Maybe if you can contact me, I can bring you some contact with that linkage and how they run it. Maybe you can invite them and give a talk on this dairy waste as well. So let's see. Yeah, that's quite interesting topic. But I don't think they have any big problem for my understanding. Last time we have a conversation with them. So definitely dairy waste will be a target as well.
Thank you. So Sonia has just suggested another webinar, so good, stack them up. We need to get a nice schedule in, so dissolved methane recovery being a potential future topic. Yeah, Louise Byfield has suggested that you showed a slide on emerging contaminants. Could this technology provide benefits for a way forward for removing some of these? And I know that a lot of the water industry colleagues in the room are thinking along CIP3 chemical extractation program and how we're going to manage to treat some of these emerged micropollutants down to very, very low levels. So Henry, any thoughts on that?
Yeah, so I actually have one PhD student working on SMX with the same process, and from the lab we do see some removals because we're still building out the biofilms and want to understand how few like SMX, how they can remove that, but also antibiotic resistant genes, would that be occurrence very quickly? You remove it, of course, but you create another problem but for resistant genes, so we're still doing that as well. But I'm sure this is quite robust and they can remove those, but then you remove but you create antibiotic resistant genes. That can be another challenge. So we are towards more on the resistant gene as well, not only bio removal in those pharmaceuticals.
I guess it's a small price to pay if you get effective biological removal, so you're not discharging to the environment. Great, okay. And then we've got one from Vicky saying, did the lab scale have pH control with fixed or variable pH? And if so, no. Is there any observable effects of pH on the biofilm?
No, because it's good. The system is really efficiency. We barely measure VFA, so the pH will not be easy. It will be easy to control. So we're never able to control the pH. That's a beautiful part of the systems, because you got bacteria and they like the food. You know, with Geobacter and Methanotrix, they all hang it up for the acids, so the pH is the VFA is so low, so pH will not have a big issue in the systems.
Fantastic, thank you. And I guess an interesting point is this raises the prospect of using a technology like this, so conductive materials and diet in something like a drinking water treatment works for NOM or pesticide or something like that.
Yeah, that actually I also reached some of try to reach some of drinking water companies. If they have GAC, they have biofilm, I can willing to do some sequencing for them and see is this diet in that system. If any water company interested that, we can talk about that. We have this kind of idea, but haven't, because COVID, we haven't really done anything on that. But this will be interesting to see drinking water part on this diet.
Yeah, well, there's lots of colleagues on the call, so I'm sure if they've got any capacity for that, they'll reach out directly. So we're getting to the end of the time. I just wanted to firstly thank Dr. Lee for an amazing presentation and starting off this water biofilm series. So a virtual clap.
Oh, thank you so much.
One day, hopefully, we might be very good. Thank you, everyone. We might be able to do these in person, hopefully. We are also looking at trying to arrange a biofilm linked conference, so this will be specifically focused on water biofilms. So watch this space. That would definitely be an in-person or hybrid event. So there's an opportunity for us to get together again and learn a bit more about biofilms. So with that, I'd like to close the session. Any questions or further comments, please email Dr. Lee directly about his talk, and I'm sure he'd be willing to carry on the discussion. Anything to do with water biofilm seminar series, please do get in touch with EBNet directly or myself, and we will get things moving. So with that, I look forward to seeing you all in September, where we're going to hear from Professor Joby Boxall on biofilms in drinking water distribution and storage systems. So with that, I'd like to close the session. Thank you very much.